Every year during Brain Awareness Week, we've had a public event of some sort. But this year, at the urging of one of our new faculty members who joined us from Harvard about a year ago, Decarli Fiorante at the back, she encouraged us to try to do something a little bit bigger and better, and to do it here at the center, bring the public to the center. Our center is an interdisciplinary center, and it houses roughly 25 laboratories where we investigate the function of the nervous system, the brain, and try to develop our basic fund of knowledge so that we can not only understand this amazing, miraculous biological machine, but also understand how it becomes perturbed at times. It goes wrong, it's not perfect, and often leads to disorders of the nervous system, brain disorders that are familiar to all of us. And we're working very hard to understand those disorders as well and to try to find cures for them. Before we start, I want to give some acknowledgement firstly to our speakers and to our graduate students who have so generously given up their weekend to come and to help to educate you about their particular areas of expertise. And we'll be talking about 4 different kinds of brain disorders today, and the neuroscience, the basic science that underlies them. I also very much want to thank Decarli Fiorante, who I just introduced you to, and Karen Zito, who are our sort of local Brain Awareness Week leaders who organize a lot of events. In addition to this particular event, our graduate students go out to many schools as well as to the farmers market and take the excitement of neuroscience. They take their booths with their hands-on experience out to students to get them interested and excited about science. And they do this work, actually it goes for more than a week, and they generously give their time, so I'm very grateful to them. I also want to thank our development officer, Jennifer Scott, who's up the back in the blue jacket. She's also played a big role in organizing this event. If you sign up to the sign-up sheet, and if you're interested in finding out more about the center, Jennifer's the person for you to go to. I also was instructed to tell you before we begin where the bathrooms are. Those are out the door and to the right. Men's first, and if you keep going, you'll come to the women's bathroom. We've got a full afternoon of talks and events, and then we've got a little reception at the end. We've got some cookies and things for you to eat. And I'm just going to go ahead and introduce our first speaker. So our first speaker is Professor Charlie DeCarli. Charlie is the director of 2 Alzheimer's centers that we have here at UC Davis, both the federal and the state ADC. Charlie is a dear colleague of mine. He was actually my neighbor when I moved back to Davis 12 years ago, but after I moved in, all of a sudden he got up and left. However, we remained friends and colleagues. So Charlie is one of the national, international leaders in Alzheimer's disease. He's an expert in non-invasive brain imaging, particularly the use of MRI, but also positron emission tomography, PET imaging, to better understand both healthy aging, the things that we can do to help our brains stay healthy, as well as the whole range of conditions and disorders that can lead to late life cognitive decline. So I won't say more, I'll just ask Charlie to begin.
Thanks a lot, Cam, and I really appreciated the opportunity to talk to you guys today. Louder. I really appreciate the opportunity to talk to you. Thank you. My definition of my work is that I am a translational cognitive neuroscience scientist, and that means that I study, I take the knowledge from all of the young people who are working on the primary understanding of how the brain works and investigating all the exciting new technological things, and I apply it to what is loosely termed as diseases of aging. And in fact, there are some of my colleagues who feel aging is a disease, but I think of it as a process in which we refine ourselves like a good wine. You know, we're getting better with you. Of course, that means you drink red wine and not white. So, but anyway, my job is to understand how, using the tools that they have here at the Center for Neuroscience, applying these tools in a way to understand how the brain ages and then what happens when there's disease. So one of the things I'm going to talk about, if this works, I'm going to give you a basic tour You saw that little video. I'm going to recapture some of that video about nerve cells and nerve circuits, how to measure the brain, what's normal aging, the structure of the brain, and then common diseases of the brain. Okay? And I'm going to do that because I'm going to try and help you understand that the brain is a complex organ that changes over life, but there's really some good stuff that comes out of that as we get to understand it better. So, you saw that pretty picture. I didn't quite have that kind of creativity in myself, but we're going to talk about cells in the brain. Cells, most of the cells in the brain are not nerve cells. They're actually supporting cells, but they're what we think about as the brain structure and function is related to a nerve cell. It has a cell body. It has these little dendrites that carry the signals, and then the axon that sends the information that the nerve cell gets and kind of works on and integrates, and then sends along to communicate with other nerve cells. And this is a cartoon picture, and this is a real live picture of a nerve cell, and you can see that we're getting very, very, very good at taking pictures of nerve cells. And in fact, some of my colleagues are going to show you stuff that's just going to blow your mind about how we can look in the nerve cell and see how it sends packets of information, sends all kinds of things along, and how it regulates itself and develops. One important feature of nerve cells is that they're very social. They tend to hang together, okay? So you put a nerve cell in on a petri dish, it tends to grow all kinds of other little nerve cells, and they want to connect. Their job is to connect to each other. They're sort of like social butterflies. It's important to understand how the brain works, to understand that the nerve cells in a normal brain connect in patterns. So they connect in specific patterns, and these specific patterns help to integrate and make function, okay? And so even within a part of the brain, the gray matter part which holds the nerve cells, there's all kinds of connections that help us to put it together to make it work in a specific way. Okay? So they're talking to each other, but they're talking to each other in an organized way. Okay? This is very much unlike a group of Italians in a dinner meeting who are all talking to each other. It's more like the Germans. Somebody says, I have a question, and then everyone says, okay, ask the question. So they're much more organized. Okay? Importantly, there are these, not only do they organize within one small part of the gray matter, but even within certain parts of the gray matter, they begin to form a circuit, okay? And this is like a kind of like an integrated circuit that you think about when we think about our cell phones and things like that. If you ever open the back of these, you see those little things that are, you know, the green with the little lines on it. Well, this is the kind of stuff that happens in certain areas of the brain. And the reason that these local areas are connecting is because they're making specific functions. Okay? So every part of the brain has a specific function related to it at that local area. But then, as you'll see in just a second, it shares that information. So the brain is set up with a bunch of different areas. Now, I'm not going to go into this at all, but what I want to say is that the brain is, so we have organization at the microscopic. We increasingly spread out this organization. So it's sort of like we've got a little circuit here, then we build that little circuit here, then we add on to that circuit, and we've got a little like a transistor. And then we're going to have another transistor over here, and then we're going to have another transistor over here. And the way this works is that the brain actually has different cell organization throughout the brain, and these different cell organizations help to make the brain function differently. And the best way to think about it is you start small and you build up and you highly organize it so you get to a place where we can then understand some of the things that we talk about commonly, like speech or vision. Okay? And you'll hear more about that later, I believe. Okay, now, I do this because I'm so self-deprecating, but it's loads of fun, okay? Cold hands is a big part of the brain of a doctor, yes. Fear and loathing of lawyers is also another big part. The point here is that there are circuits that work to have certain functions, okay? So there is a part of the brain that has to do with speech, and I'm going to show you that in just a second, and hearing. But that doesn't make us who we are, okay? So what happens is that we have these local circuits, and then these local circuits are stuck together in larger circuits, which we call brain networks. Okay? So here's a part of the brain up front here that controls how my mouth moves when I make speech. Okay? And it's very specific about controlling how my mouth moves. Okay? It doesn't do much else than that. All right? But it receives information from back here and all other places, and it controls how my mouth moves to make the speech. But just knowing how to make speech isn't going to help me unless I can interpret speech. So this is the output part of the network, and this is the receiving part of the network. And this is a part of the brain that is specific to understanding speech. So information comes in through your ears here, it gets processed, and it says, oh, okay, he's making a sound. Not just any sound, he's telling me something. And then he asked me a question. Okay, so that goes over here, and I have to respond. And I say, do you like red? No, I like blue. Whatever, okay? And these circuits that you're seeing, these networks, are connected by white matter tracks, okay? And those are those axons that are coming out from the nerve cells. And I think of this like the cables on telephones, or if you've ever seen, back in the old days, how they had those phone banks where they're all connected, same thing, okay? All this information comes through long wires, and these wires are connecting things to each other. And they're well named and well understood, and there's local connections, and then there's big, long connections. So the brain is formed of nerve cells. Nerve cells form connections. Local connections help develop very specific parts of the brain, okay? Like this part that moves my mouth, okay? But these things are then connected into networks that are the building blocks of how we think. Because not only am I talking to you, but I'm perceiving things. I'm making sure that none of you are falling asleep. I'm doing all kinds of stuff, okay? All right, so I'm not just using one part of my brain. I'm probably using 2% instead of We can measure the brain in life, and this is something that started, I'm kind of an older guy, but that gives me a historical perspective. When I started all research, they had CAT scans, okay? Now we've come of age where we can have very sophisticated tools to look at the brain. So we can look at the brain structure using magnetic resonance imaging, brain connections using diffusion tensor imaging as well. We can use brain function using functional MRI. We can also look at brain disease. So we can look at basic things as part of how the brain is actually working during life and how the shapes change during life, and also how it's affected by various diseases. And I'll talk about 2.5 diseases, one of them being related to age. Okay, here's a picture of the brain, okay? So there's various ways to take a picture of the brain. This is as though you've taken a slice down the middle, and you're looking, I can't tell if that's the left or the right, I have no idea, but assuming that we're looking from the right side to the left. You can also look at different types of tissue. So we can look at the tissue types that enhances this fluid around the brain, turns it white, that helps me to see how big the fluid spaces are. But also we can look is whether the white matter tissue that I talked to you about earlier gets damaged by using other different mechanisms, other different sequences of MRI. So we can look at the shape of the brain, we can look at the tissue contrasts as well, okay? And because we're really clever people here at the Center for Neuroscience, we can put them together and we can make really cool pictures. And when we make really cool pictures, that tells us something about the brain, okay? And the things that can tell us about the brain are, remember I told you about those specific areas of the brain that have different cell type? We can divide the brain up into different lobes. We can also get down to very small regions that have more specific function. This is one of my favorites. Dorsolateral prefrontal cortex is one of my particular favorites. It doesn't matter to you guys, but it's something that I'm interested in. Not only can we show the size of a particular brain area, we can also show the thickness of the gray matter part, okay? So the brain is divided up into 3 tissues, gray matter, white matter, and fluid, okay? And the gray matter has the nerve cells, the white matter has those axons, and the fluid is just to keep us so that if we walk down the street and bump into a tree, we don't crush our brain. Woodpeckers, right, they're beating on, they have more fluid around there because it kind of protects the brain, and it also bathes the brain in nutrient fluids. So we can also then, over time, look and see if thickness changes or if there's expansion in these things. So we can do some very dynamic, very clever things with the brain. This is something that my laboratory does a lot of work with. My laboratory also does a lot of work with what's called diffusion tensor imaging. I talked to you about the gray cells. Well, this is a way of looking at how axons, how the connections are working, and you can see that these connections come from the gray matter tissues and then connect to other parts of the brain. And what's really unique is that we can actually, just like the electrician does in your house, where, if you've ever had this experience, where they're trying to figure out where the break in the system is, and they put those little things and they can measure how the resistance is, we can measure the resistance of these things to look at how healthy they are. So we can do this, and we can do it at a track-by-track basis, and you'll see a little bit of that data. The last thing I want to show you about MRI is we can do functional MRI. And this is really cool, and there are a lot of people here in the center who are doing this, and in the Center for Mind and Brain. Basically, a nerve cell needs two things to operate, only two: sugar and oxygen. That's it. Okay? Yeah, that doesn't mean to run out and eat a bunch of sugar.
I would say that.
I knew somebody was going to think that. And so we can, by a unique property of red blood cells and a magnetic resonance imaging, we can measure how much oxygen is being used by nerve cells. Okay? And this is very, very dynamic. I'm not going to show you actually how dynamic it can be, but this is somebody who's tapping their hands, and you can look at how the blood flow changes when they just tap their finger. And this can be extended to all kinds of tests, very sophisticated tests. Some of the tests we're really interested in is how the brain uses different amounts of information. And again, specific parts of the brain do specific things under these conditions. Finally, positron emission tomography. All of you guys who are Star Trek fans might have heard about this positronic brain in data, but that's actually, positron's a real thing. It's a mutual annihilation of a gamma ray. And what happens there is you get a small dose of a radioactive substance, very, very tiny dose, and that when that radioactive substance goes into your brain, it releases that, and we can measure that using a very fancy system of sensors. What we can do with that, which is very important, is we can look at how it uses sugar in the brain, and we can look at that in diseases. We can also label pathological processes like Alzheimer's disease and show whether it's present or absent. So we can use this information. I'm going to finish the rest of this talk now to just sort of focus a little bit on how MRI can image the brain and then some of the age-related changes that we see in the brain. Okay? So we can do the as I showed you, the structure of the brain. We can do size, shape, thickness, whether it's tissue injury or not, the function of the brain. PET can look at the sugar use, which is the function of the brain, and the pathology of Alzheimer's disease and other things that we might hear about in my colleague's talk later. Okay, so what happens when you get older? Oh, am I not talking loud enough?
They wanted to.
Okay.
I don't know this. Oh, wow. This is the time where I wish that I was Tony Bennett. Okay, let's see if we can do this.
Is that better? Oh, Jennifer's saying, yes, that's better. All right, so let's talk about how we age, okay? The way we think about brain function and the way we think about cognition or thinking, okay, is that we develop skills that equate to some kind of cognitive ability. Now, one thing I'm going to tell you is that there is no single measure of cognitive ability, and in fact, IQ sucks. Okay? You know what IQ measures? The socioeconomic status of your parents. Okay? I should know that I have 109. No. But, okay, so, you know, we just can't, people use, you know, it's all this stuff about education and stuff, people try to use all these numbers, and we're not as good in math and all this stuff, and some of that may be true. We may be terrible at math, okay? And that I think we need to, you know, there are many reasons, my wife's a teacher, there are many reasons why I think we need to improve our educational system. But going to bean counting isn't one of them. All right, so we develop some kind of ability, and then we get to some zenith of our cognitive ability, okay? And then the aging process takes over. The ideal is that we maintain the zenith of our aging cognitive ability, we're playing tennis, and we drop dead. Now, it's a little disturbing with your tennis partner, but man, it's really good for your life. Okay? But we know that there are changes, and I'm going to go over that with the brain as we age. Okay? And the important feature is to understand that these are under influence about a whole bunch. And one of the things that's really unique is we've always thought, okay, you're given this genetic ability. I'm not going to talk about this, but I think it's an important thing. You're given this genetic ability that has to do with how much you get up here. It has nothing to do with how you do out here. That is absolutely, completely, 100% false. Okay? We just don't understand it. There are lots of influences out here that relate to genetics, and we're trying to understand that. And part of my job actually is to work on that. But things that you don't think about, like nutrition in early life or early life environment, education, of course, we all think about this, but it's important to understand it's the quality of education. Okay, you can count backwards from 100 by sevens, and that everybody thinks you're really smart, but that may not be all you need to do. Other thing that I'm going to talk about are medical, what we call medical comorbidities, your medical problems that you have as you get older. Common ones. A little bit chubby, hypertension, diabetes, high cholesterol, heart disease, blah, 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 blah. Finally, interestingly enough, is your social network, your exercise, creativity, things like that. How engaged you are in your socialization is very important, how your brain maintains its ability. I'm not going to talk about cognitive reserve, only to say is that some people have every black mark against them and do really well. Okay? So nothing I say should be taken as God's truth. Okay. Here's what happens when we get older. If you take a bunch of older people from the age of 65 to 95 and you ask them to remember a story, and then you ask them how many bits of information they recall, what you see is this nice line. It says, oh, as you get older, you're remembering less of the story. Okay? Very important point. This is what the population does. This is what individuals do. Okay? My favorite is this 90-year-old who gets a lot better at age 30. And it's not just because you're doing below average that this is happening, okay? People are improving even when they are a little bit below average. Okay, but there is something going on here in a group of them that is really falling off the curve. And when you do this regression here, there's something going on, and this suggests that there might be something about disease. Not every brain function changes with age. And in fact, our memory, our performance over age of the world knowledge, okay, our world knowledge, our language, actually our vocabularies grow throughout our entire life, if we let it. But other things are hitting the skids, okay? Particularly motor speed. Anything that has to do with motor processing. That's why you're zooming down the freeway and you see somebody who's about 163 years old and they're going about 10 miles an hour. And you're like, because they know, like my dad told me when he was getting up there, that, you know, I just don't have the reflexes I used to. Okay. So now we're going to transition a little bit into talking about some of the diseases. But here's what I want you to understand. There's a lot, so you have this ability, you have things that happen over time. there's a lot of stuff that happens before any obvious disease, and this is called brain injury, and we don't always understand it. So when I'm talking about aging, it's very hard sometimes to talk about how does the brain age when there's no disease, because there's almost no one with no disease. And in fact, they've taken to people who are 80 years old with no disease, no recognizable medical illnesses, as superagers. Okay? So it's really interesting how we define that. Okay? So aging changes, it's got different things that are going on in the brain as we get older. There are many different influences, and I didn't talk about this, but the kind of speeded and frontal part of the brain is the one that changes the most. Okay, how does the shape of the brain change when we get older? Most of my work, I'm a Framingham Heart Study investigator. For those of you who don't know about the Framingham Heart Study, it's been going on since 1948. It is a community-based study. We know a lot. We have, this only shows 2 generations. We actually have 3 generations of people in the original cohort, the offspring study, and the 3rd generation who are quite young right now. The point is that we get to actually model how the brain ages in a normal population. Normal being a community. So this would be like going to a city of Davis, slightly bigger than Davis, and asking everybody, do you want to have an exam every 2 years for your entire life, and doing a bunch of tests on them. And what we see is that the brain, the whole part of the brain, is pretty unchanged until about age 60, 62, and then starts to decline slowly as you get older. Okay? These are disease-free people. These are people who come into the office and who are completely, for the most part, completely independent. Different parts of the brain age differently. The frontal lobes are taking a big hit over time. The front part of the brain takes a big hit over the time. And unfortunately, men just never get it right. Of course, my wife would totally agree, but it's true. Other parts, the other part, and this is really important because I'm going to get to this to show you, the other organ in the brain that's very important, the memory organ. So the front part of the brain is related to how we think and how we process information, and how we make decisions, how we plan for the future. Our memories, which says, you know, what did you have for dinner last night, okay, is in the hippocampus, and it too changes over time. And it begins to change in about the same time, 62 to 65 years of age. And again, men tend to have smaller, they start off bigger and then they get smaller. And those were what we call cross-sectional, so I sort of associate that as the aging process with a bunch of different people at different ages. And my colleague, Naftali Ross, has actually said, okay, let me look at 50 or 100 people over time, and he shows the same exact pattern. And Randy Buckner's done this, a bunch of people have done this thing longitudinally. Now, we also know that the integrity of those white matter tissues change with age too. So not only is our brain shrinking, but its connections are changing in a very subtle way. I have to say our tools are so sensitive that we can see very, very subtle changes that are highly significantly significantly associated with age, but they really may not be all that meaningful. But to say is it's not only the gray matter that's shrinking, but the white matter connections are changing. And I just did something bad. What did I do? It's not my fault. Oh, this little thing you tied? Oh, maybe there we go. Okay, and that this changes in different regions, okay? So not every region, just like in the cortex, in the white matter, not every region changes the same way either, okay? So it's a very complex bunch of changes in our brain. The last thing is that even the function of our brain changes, and one of the common things that they talk about is that as we get older, our ability to perform some of the tasks that we do actually may even improve slightly, or at least stay the same, but there's a consequence. We need more parts of the brain. So we tend to have, young people tend to use only a little part of their brain, sort of like cleaning up their room. They don't pay much attention to it. They only need a little piece here. And so, but older people, and this is a group of about 70-year-olds, need both sides of their brain to accomplish the task. And it really doesn't matter which the task is. This tends to be fairly typical across all tasks. memory for lists, or working memory, where you're trying to process things in time. So typical things are you're asked to do some kind of manipulation of information and then respond to that. Oops. So, age-related differences in brain structure are large. Measures of brain structure and white matter integrity are regional, and some compensation may occur. And in fact, I think that a lot of compensation occurs. Okay, so that the brain maintains its ability despite these age-related changes. And we just don't understand why that happened. But understanding how those age-related changes occur will help us to be resistant to disease, which is what I'm going to talk about now. So, we worry about things like medical comorbidities. And I just want to point out that getting older is commonly associated with all kinds of problems, particularly hypertension. Hypertension is a risk factor for stroke. Stroke and Alzheimer's disease are really common, when we get older. Unfortunately, stroke is the tip of the iceberg. So I don't know how many of you know about stroke, but that's where suddenly you get weakness in the arm or leg, or you have problems talking, and it's due to the occlusion of a blood vessel going into the brain and really injury to the brain. But that's just the tip of the iceberg of things that can happen to the brain. So you can get small, tiny strokes, and I'm sorry, I forgot the picture. You can get those white matter changes. I showed you way back when, where I showed you that we can look at the tissue integrity, and we showed those blotches on that one scan. Okay, right? This relates to vascular risk factors like hypertension, diabetes. So does this. And then hypertension can cause your brain to age faster. Okay, so it kind of like, I don't know if this analogy worked for you, but people who do too much sun, they get the wrinkly and they look older than they're supposed to look because the sun kind of, I don't know what the heck it does. That's like, it causes wrinkles. Well, hypertension causes wrinkles, but only in your brain. Okay, so that there are things that out there that are causing our brain to age prematurely, and we have to understand those things and prevent them. And that these things are common, and that they cause our brain to age, and unfortunately, when our brain ages, so if, I want to be a 60-year-old, I want to have a 60-year-old brain. I don't want to be a 60-year-old and have a 70-year-old brain. Because my biggest concern is that if I'm 60 and have a 70-year-old brain, what's my brain going to look like when I'm 70? Not so good, okay? And vascular disease is one of the primary movers for that. And so the best way to get around that is to have a healthy lifestyle and to treat all your problems that you may have, particularly hypertension, obesity, and diabetes. Okay, there's a second disease that happens as we get older, and very common, and that's called Alzheimer's disease, and we have ways to look at that. And here's the idea, is that Alzheimer's disease is the result of an abnormal protein called amyloid beta, and it forms plaques in the brain, and that these plaques then do other things to the brain, and that leads to forming what are called neurofibrillary tangles, and those neurofibrillary tangles are, in essence, dead nerve cells. And you don't want dead nerve cells, okay? And this leads to dementia. And the idea is that the amyloid accumulates years before we ever have any other consequences, and that, in fact, we can measure some of these secondary pathologies due to this amyloid accumulation, and then we come up with behavioral problems, nursing home, and death. Okay? So this is the pattern of Alzheimer's disease. My colleague, Bill Klunk and Chet Mathis discovered about 2004, 10 years ago, 11 years ago, how to measure this in the living human brain. And here's a picture using their particular thing of someone who has Alzheimer's disease. This is actually a brain slice. And then this is what it looks like in a photograph, okay? And what they discovered almost immediately is that there are normal people who have no amyloid in their brain. There are normal people who have amyloid in their brain. There are people who have cognitive impairment who have no amyloid in the brain. And there are people who have mild cognitive impairment who have amyloid in the brain. Everyone who has Alzheimer's disease, by definition, has to have amyloid in their brain. So right off the bat, it's a really good diagnostic tool for Alzheimer's disease, because you can't have Alzheimer's disease without having amyloid in the brain. But importantly, you may have amyloid in the brain and not have Alzheimer's disease. You may not have the dementia of Alzheimer's disease. And what we see, if we take a population of normal people, is that as we get older, we have more and more of this. And in fact, by the time you get to 80 years old, almost a third of people have this in their brain. They're not all demented. And in fact, this is 200 normal people. So we don't quite understand how you move from having the amyloid into your brain to forming the dementia, but we at least have an ability now to look at this. So we can look at the cognitive neuroscience tools, and the neuroscience tools we have now can look also not only the structure of the brain, not only how the brain operates, but certain pathologies in the brain and how they influence these processes. And so we get, oh, sorry, forget the genetics. Okay, so, yeah, it's hot in here. And I'm wearing a suit coat.
Take it off, the jacket.
Take it off. Did I hear that right? Yeah. I'm way too old for that. Okay, here's what I want you to understand. As we get older, our brain changes, okay? And it changes in many different ways, okay? But for the most part, we are able to compensate for those changes if we remain healthy. Neuroscientists are beginning to identify diseases years before they affect our brains in a way that is clinically significant. And that what we're trying to understand is how does the brain age normally, and how those first steps of the disease can be modified so that those two processes can be. So basically, what do we want to do? My wish is that we all have healthy brains and we live till we get to 95 playing tennis and drop dead. And if that happens common enough, then nobody will worry. They'll just cart you out and everything will be fine. Okay. It's influenced by a whole bunch of stuff, and I have no problems knowing that the rest of my career is fully going to be busy, because every time I turn the corner, I find new questions and that are in need of answers. And so understanding how the brain ages is important to how it suffers with disease, and understanding how those two processes occur helps us to maintain healthy, happy lives. And I'll thank you very much.